Hi, I hope you're doing fine. So we were studying biochemistry. 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 Interesting subject. Boring? No, it's interesting. So let's try to understand what I have discussed so far. Just in one line, that food gets energy from the sunlight and the carbon dioxide, while we extract energy from the food by its oxidation. Then we discussed in the second last video that how food gives energy. I introduced 5 steps. First was digestion, then absorption, then formation of acetyl-CoA, then Krebs cycle, and finally the electron transport chain. This you should know. But now I am going to give you an overview, big picture. Yeah, break baat sirf. Just one thing that I make you to understand. Although these things are not going to come in your exams, they will never come. But these things are the backbone for your studies. If you don't know this big picture, it will be really difficult to integrate different concepts. But where is glycolysis? What is the purpose of glycolysis? What is the purpose of gluconeogenesis? What is the purpose of glycogenesis? And all those pathways. If you don't understand this, biochemistry will be like boring cycles, pathways, and we can never understand them. We just have to cram them. But if you understand the big picture, what's happening why these things are happening, then you will be very much interested in understanding all these things. So this is investment, just investment in understanding the basic concepts. And if you have invested right amount of time in understanding these basic concepts, then you will be on highway and you will be riding like 120 kilometers per hour. But first you need to run slow. So today I am going to discuss what is the overview of energy metabolism. What happens in the different parts of our body during the well-fed state, and in the subsequent video I will be explaining what happens in the body during the starving state. So let's start. Are you with me? You're yawning? No? Oh, great. Okay. Now, come to me to understand this. You see that? Oh, this whole thing is your body. This page is your body. Can you imagine it? This page is your body. Huh? Yeah. And you imagine this also, that this which I am drawing is liver. Okay? And this I am drawing is brain. And this I am drawing is red blood cells. And this I am drawing is muscle. And here is the adipose tissue. I'm not an artist, but biochemistry is an art. And when I say that this is muscle, I don't mean that this is muscle. I mean that muscle cells. When I say this brain, I mean that this is the not brain, वो जो बड़ा सा नजर आता है जिसमें parietal lobe भी है, frontal lobe भी है, temporal lobe भी है. I'm not talking about this. I'm just talking about the cells of the brain. Similarly, if I talk about liver, it means that they are the hepatocytes, not the liver per se. And similarly, adipose tissue means adipocytes. Alright? Yeah. So understanding things in metaphorical terms is essential in learning process. That you should know what is the meaning of it. Okay? Muscle doesn't mean muscle which we see like red muscle or white muscle. I'm not saying that. Muscle, I mean to say muscle cells. So all these things. And what is outside the box? Outside the boxes, I mean these spaces, you see here these spaces, all these spaces are blood. What? Blood. Now, and I, as in my previous tape, I explained that after absorption all the non-hydrolyzable nutrients. Which nutrients? Glucose, galactose, fructose, fatty acids, cholesterol, amino acids. They are in the blood. Now what happen from the blood? They are definitely going to go into the cells. And what is happening in those cells? In this video or in this tape, I am going to explain that. Fine? You see that. So here, for instance, we have glucose. And that glucose goes to the liver by crossing the cell membrane of the cell, and here there is an important transporter which is called glucose transporter. I will explain this in detail, what are glucose transporters and what are their types, when I will be discussing glycolysis. So that glucose which is now in the blood goes to the liver. And from our previous overview, it's the glucose is going to be converted into pyruvate. And pyruvate is going to be converted into acetyl-CoA. And acetyl-CoA into carbon dioxide in Krebs cycle and ATP in electron transport chain. You see? And this all happens under the influence of insulin. You know, we are in well-fed state. We are in well-fed state. खा पी के रजे हुए हैं. And when we are in well-fed state, there is lot of glucose in our blood. Lot of glucose in our blood. And that all glucose need to be internalized into the cells. सारा का सारा ग्लूकोज किधर जाएगा? सेल्स के अंदर, अंडर द इन्फ्लुएंस ऑफ इंसुलिन. सो इंसुलिन डिक्रीजेस ग्लूकोज लेवल. हाउ? ग्लूकोज लेवल इन द ब्लड. ग्लूकोज लेवल in the blood. How? By pushing this all glucose which is present in the blood to the cells. If there is no insulin, this glut will not be working, and if it doesn't work, the glucose will not enter into the liver cells or in the red blood cell or in the muscles or in the adipose tissues. Okay, so what is the purpose of insulin here? Purpose of insulin is to decrease glucose level in the blood by pushing the glucose from the blood into the cells. Alright, so glucose first enters into the hepatocytes, and that glucose is converted into pyruvate by the process of glycolysis. This pyruvate is converted into acetyl-CoA, and then acetyl-CoA enters into the Krebs cycle to make carbon dioxide, and then finally the electron transport chain produces ATP. This is the first step. This is the first step that glucose is going to take in the liver cells. And once the cells meet their energy requirement, sufficient energy has been produced, then the extra glucose is going to be converted into glycogen. Okay? First, the cell meets its energy requirements. Okay? And once the energy requirements are met, the extra glucose that is entering into the cell will be converted to glycogen. Okay? Now when the glycogen stores are full, then we still have acetyl-CoA. We still have acetyl-CoA because a lot of glucose is coming in. We have eaten a lot of starch in the form of biryani, in the form of potatoes, in the form of flour, bread, in the form of pizza, shawarma, all that stuff. We have eaten lot of starch, and all that lot of starch has made lot of glucose, and all that glucose is in the blood. But we need to transport that glucose from the blood into the cells. The first cells do what? They make ATP. And in the second step, they make glycogen. They make glycogen. Clear? Right. Now, glycogen store is also full, but glucose is still entering. So this extra glucose will make acetyl-CoA. But now acetyl-CoA will not make ATP because ATP is already full. So now this acetyl-CoA makes fatty acids. Okay? And also cholesterol in the liver. So actually there are 4 things that glucose can do in the liver. One, which is that glucose is going to make energy. Second, the extra glucose is going to make glycogen. And if still glucose is entering into the liver, that extra or excess glucose will make acetyl-CoA, and then that acetyl-CoA will make fatty acids and cholesterol. That is the reason when we eat carbohydrates, all those carbohydrates, extra carbohydrates, are stored in the body in the form of fatty acids and cholesterol. Okay, so this is the first thing. I hope you understand. 4 pathways that glucose which enters into the liver under the influence of insulin in well-fed state is going to do 4 things. First, it will meet the energy requirements of the liver cells. That is to say, it will make ATP. And if sufficient ATP has been produced, the extra glucose is going to make glycogen. And if the glycogen stores are full, then the acetyl-CoA, which is formed from the excess glucose, will make fatty acid and cholesterol. Clear? याद हो गया ना? Did you learn it by heart? Learn it by heart. Please learn it by heart. It's essential. Glucose makes first the energy, then the glycogen, then the fatty acid, and simultaneously the cholesterol. Clear? Fine. Now, the same glucose goes to brain, again by a transporter which is called glut. And that glucose will make what? Pyruvate. Yes, good. And that pyruvate is going to make what? Acetyl-CoA. And acetyl-CoA is going to make what? Carbon dioxide through Krebs cycle, and ATP through electron transport chain. An important thing, brain is very, very hungry of glucose. It is living only on glucose. It doesn't like any other thing. It just likes glucose, all the glucose. And this glucose must be provided to brain under every condition. Alright, glucose is essential for brain. Only glucose. And 25% of cardiac output, 25% of cardiac output is received by brain. 25% of cardiac output is received by brain, right? And that means that why? Because it needs lot of glucose. If glucose is not provided to brain, it is going to shut down. That is the very reason hyperglycemia is not dangerous. Hypoglycemia is very dangerous. If you have low glucose level in the blood, brain will not get glucose and it is going to shut down. And that condition is called hypoglycemic shock, which may lead to coma. Okay, so important thing that brain lives on glucose. Now we come to the red blood cells. Same story, but a little difference. Glucose makes pyruvate. And as I told you in my previous tape, that anything that happens after pyruvate happens in mitochondria. But here in red blood cells, we don't have mitochondria. So that pyruvate is not going to make acetyl-CoA, because for formation of acetyl-CoA we need mitochondria. That pyruvate is going to make lactate. Why? That I will answer in glycolysis. Now this lactate from the red blood cells goes to the liver. And in the liver it makes again pyruvate. And this cycle is also called Cori cycle. Okay, what is Cori cycle? The transfer of lactate from the red blood cells to the liver and then its conversion into pyruvate. That is from pyruvate, its journey from pyruvate to pyruvate. Okay, pyruvate makes lactate, lactate enters to the liver cells, and then lactate again makes pyruvate. Right, this is the story in the red blood cells. Now what happens in the muscles? Same glucose enters to the muscles through glucose transporters. Now this glucose is going to make what? Now you should tell me. Pyruvate, yes. And then this pyruvate is going to make what? Acetyl-CoA. And then acetyl-CoA is going to make what? Carbon dioxide and ATP. Okay? And similarly, the same glucose goes to the adipose tissue. Makes what? Pyruvate. But adipose cells, adipocytes have mitochondria, so pyruvate is going to make acetyl-CoA. And acetyl-CoA is going to make what? Carbon dioxide and ATP. So that was the story of glucose. I hope you now understand. Now, what about amino acids? I'm referring amino acids by AA. These amino acids will go to muscle cells. 3. So we were talking about amino acids. I'm referring here amino acids with AA. That amino acids enter into the muscle. What is going to do here? Amino acids are going to make protein in the muscle cells. Okay? And the same amino acid goes to liver. And it removes its amino group. It removes its amino group and make urea. While rest of the things make pyruvate. And this is called urea cycle. Alright? So that is the whole story with amino acid. Right? Now there is one thing left, and that is lipid. You know, I explained you that from the diet what comes in the blood circulation? Not fatty acid, not cholesterol, but chylomicrons. You remember? Chylomicrons. And then these chylomicrons then break up and they release their fatty acids. They release their fatty acids into the adipose tissue. And in the adipose tissue, this glucose makes glycerol as well. And then glucose and fatty acid combine to make again fat. And that fat is stored there until it is neutralized, or it is used for nutritional purposes. But if there is no nutrition or nothing is required, this fat remains there in the adipose tissue. Okay? And these the other thing of the chylomicrons is glycerol. That glycerol goes to the liver. And this glycerol combines with fatty acids to make fat. And these fatty acids are already produced by the liver. This is the important point to ponder, please. I want your attention here. A glycerol, which is coming from chylomicrons here, combines with endogenous fatty acid, the fatty acid which the liver has made itself. The fatty acids combine with glycerol to make fat. And then this fat exports out of the liver, not in the form of fat, because fat independently cannot go to the blood, but in the form of VLDL, very low density lipoproteins. So the lipid that comes from the intestine is chylomicron, and the lipid which comes from the liver is in the form of VLDL. And again, VLDL is going to dump its fatty acid into the adipose tissue and glycerol back to the liver. So essentially, if you are with me, you see that you have taken a lot of glucose in your diet, and that lot of glucose is going to make acetyl-CoA. That lot of acetyl-CoA is going to make lot of fatty acid. That lot of fatty acid is going to combine with glycerol, and this glycerol making fat, and that fat is making VLDL, and then VLDL is going to make again fatty acids, and then those fatty acids are going to be dumped in adipose tissue. So actually, and then this fatty acid is going to combine with glycerol, and this glycerol and fatty acids are going to make fat. So I wish you to see that glucose is actually being converted to fat, which is going to be stored in the adipose tissue. And there is the answer. The people who take lot of carbohydrate diet, they have lots of adipose tissue. They have lots of fat over their belly, over their tummy, over their buttocks, all over the body. So this is the overview of energy metabolism. I wish that as you heard this all, listened this all, watched this all, watch it again, but this time with the copy. Whatever I am drawing, try to draw with you as well. Okay? And this way, this will be very okay. And this is really very interesting. This is very essential in understanding that what happens to glucose in the liver, what happens to glucose in the brain, what happens to glucose in the red blood cells, what happens to glucose in the adipose tissue and in the muscles, and similarly what happens to amino acids, what happens to the chylomicrons, the dietary lipids, and what happens to VLDL. And what is VLDL? So VLDL, here that's a point to be noted. Fats are synthesized in the liver. Fats are synthesized, fatty acids are synthesized in the liver, but liver don't use fatty acids for energy purposes during the well-fed state. All that fatty acid is going to be dumped into the adipose tissue. Alright? Thank you so much. And here was final things. Understanding this is essential. I'm just saying this for the third time, and there should be some meaning in it if I'm saying it in third time, because in this just one page I have drawn lot of cycles, from glucose to pyruvate glycolysis, from pyruvate to acetyl-CoA link reaction, from acetyl-CoA to carbon dioxide ATP Krebs cycle electron transport chain, from acetyl-CoA to fatty acid synthesis, from acetyl to cholesterol synthesis, from amino acids to urea cycle, from glucose to glycogenesis, and similarly from amino acids to protein in the muscles, protein synthesis, formation of fat in the adipose tissue, fat synthesis, formation of VLDL, formation of chylomicrons, and this whole spectrum. So I just narrated kind of some pathways, and these pathways do come in the exams, and in the subsequent videos, in the subsequent tapes, I am going to explain each of them in lot of detail, and that lot of detail is essential in understanding the chemistry of our body, and then that chemistry is important to understand pathology of the disease, pathology, and that is also important for pharmacology, because we just maneuver these steps, these reactions, to take our advantage in the form of drugs. I hope you understand. If you have any question, ask me. Alright? And whatever I am teaching from USMLE Step 1 Kaplan Biochemistry. Go and read that from there as well. Thank you. Bye bye. Have fun. Dee